various methods to diagnose infections. There are a few methods to help in the identification of bacteria from specimens and samples. Phenotypic methods look at both macroscopic and microscopic characteristics. They include structural characteristics, how they function, as well as biochemical profiles. Immunologic methods study serum analysis, while genotypic methods identify bacteria through genetics. By gathering information from these methods, a profile for a bacterium can be determined. Phenotypic methods will focus on physical observations like this microscopic view here. Immunologic methods study serum by studying antibodies and antigens in samples. Genotypic methods analyze DNA or RNA and may use gel electrophoresis in that process. As far as bacteria, genetic means are used to identify them. With medical advancement, there has been a rise in a point-of-care diagnosis where tests can be performed quickly at the bedside. CT scans, MRIs, and PET scans can also help in the diagnostic process. Phenotypic methods view physical traits expressed by an organism. It focuses on appearance and behavior. It looks at reactions that takes place in the organism, what types of environments that microbe prefers, as well as antibiotic susceptibility. This method also analyzes the components of the cell wall and/or cell membrane. Immunologic methods study the antibody response of the patient against a suspected pathogen. This response tends to be easier than trying to identify the presence of the pathogen. There are a wide variety of kits that can be used to immediately identify pathogens. With genotypic methods, you don't have to always culture the microbes. These methods are automated and rapid, and more precise results are acquired. These organisms that can't be grown in the lab can be identified using genotypic methods. The first step in the identification process is the specimen collection. It is vital that specimens are collected, handled, stored, and cultured properly so that there can be successful identification and treatment. Aseptic technique plays a big part in that success. Should a sample contain normal flora, care should be taken. This is to ensure the identification of the pathogen and not the microbiota. Only the site of infection should be sampled. There are various sites that can be sampled, including saliva, blood, urine, skin, feces, cerebral spinal fluid, and vaginal swabs. Sputum samples can be collected via coughing or via a catheter. It's important that the saliva not be collected as it contains millions of bacteria compared to the sputum. Saliva, in some instances, is needed and can be collected from expectorate. Urine can be collected using a catheter or a clean catch from the urethra. In this method, contaminants can be differentiated from pathogens. A dirty catch, which is the first voided urine, can also be used, along with swabs of the mucous lining in the urogenital region. Skin can be sampled via swabs or scrapes, along with a punch biopsy tool. Sterile regions like cerebral spinal fluid must be taken by sterile needle aspiration. It's important that antisepsis procedures are performed. Eyes, synovial fluid, and diseased tissues can also be sampled. No matter the sample site, there must be a proper collection, transport, and storage. All samples must also properly be labeled. The patient is also analyzed for signs of infection. The specimen is then collected and analyzed. The chart must be filled out appropriately with the source of the sample along with the test requested. The bottom of the chart will detail the results of the test. The phenotypic methods can immediately be examined. A stained specimen is one of the most rapid tests to determine presumptive characteristics. The stains typically used include the Gram stain and acid-fast stain. Some tests require culturing of the microbe. These methods include specialized media that allows a pathogen that's in small numbers to grow. Selected media can be used for non-sterile specimens that contain a wide variety of bacteria. It also allows for the growth of only the pathogen. Differential media can be used to differentiate organisms that have similar characteristics and fermentation patterns. A dichotomous key is a flowchart that uses test results to determine the identification of a microbe. Biochemical tests determine the presence of enzymes in a species. Typically, these metabolic reactions catalyzed by a certain enzyme are detected by color changes. The microbe is cultured with a particular substrate and then observed for an end product. The expression of the enzyme can be detected utilizing a dye. A lack of color change indicates the organism does not have the particular enzyme. Should an enzyme be present in the organism, the product formed will induce a color change, indicating a positive result. If the enzyme is absent, no product will be formed, and no color change will be seen. This indicates a negative result. This API test is able to perform several biochemical tests at once. After incubation, color changes are observed, and a numerical code is acquired. The code is specific for a particular microbe. The MGIT system can be used to identify if Mycobacterium tuberculosis is present in a sample. It monitors the oxygen levels inoculated with the sample. At the bottom of the tube is a silicone chip with a fluorescent substance. As oxygen levels decrease, fluorescence will be emitted and an automated machine will be used to determine a positive culture. Antimicrobial susceptibility is also used to determine which drug should be administered. Most automated systems include the susceptibility testing. Phage typing can be performed by infecting bacteria with bacteriophages. This method is useful in the identification of bacteria by looking for cleared areas on the media. In this image, we can see the clearing or lysis of bacteria by two different phages. It's important for a clinician to determine if what is isolated is clinically important, or if it's a contaminant. A small amount of E. coli in a urine sample can be normal, but if there is a significant amount, it could be indicative of an infection. However, if a single colony of Mycobacterium tuberculosis is seen in a sample, it is suggestive of disease. If a particular microbe is repeatedly isolated, it could mean it's a causative agent of disease. As great as phenotypic methods are, they do have their drawbacks. It takes time to culture and grow the microbe, and some diseases may be caused by organisms that we can't grow in the lab, which ends up putting the blame on another microbe. Let's review our phenotypic methods. Direct examination, like staining, does not require cultivation. Those that do include biochemical testing, susceptibility testing, phage typing, and specialized media. Immunologic methods include in vitro testing of bodily fluids for antibodies. This method capitalizes on the fact that antibodies are extremely specific for a particular antigen. This method can determine if a patient has been exposed to a particular microbe. The interaction of antigens and antibodies can be examined to confirm a disease and screen for a disease. Two questions can be asked in serological testing. The first is, does the patient make antibodies towards a microbe? The patient's serum with unknown antibodies are mixed with a known antigen, let's say syphilis, for example. If the antibodies against syphilis are not present and no binding is seen. However, if the patient does produce antibodies, a reaction will occur. The second question is, what is the identity of the microbe from a sample? A colony that was isolated from the sample is mixed with known antibodies, in this case, Staph aureus antibodies. If binding is seen, it indicates the presence of Staph aureus in the sample. Immunologic detection methods include agglutination and precipitation reactions that you just saw. They also include immunochromatography, Western blots for protein analysis, fluorescent antibodies, ELISAs, and in vivo tests. The immunologic methods rely on antigen-antibody binding. This binding is not something we can see with the naked eye or basic compound light microscope. If the complex is suspended in a solution, we may be able to visualize it as agglutination. In both agglutination and precipitation, the antibody and antigen are linked. In agglutination, antigens can be whole cells. The reactions can be seen as clumps, and this is the basis of blood typing and compatibility testing. In precipitation, the antigen is a soluble molecule that can easily be disrupted in liquids. In A, we can see a tube agglutination test that determines antibody titers. B illustrates a microtiter plate that depicts hemoagglutination. Both positive and negative controls are included. If enough antibody is present, they stick to the bottom of the well. If not enough antibodies are present, the red blood cells sink to the bottom, with antibodies floating at the top. Immunochromatography is the basis of pregnancy test and the rapid strep test. The cartridge contains a substance that directs fluid in a particular direction, which is the patient's sample. As the patient's sample flows, it encounters antibodies along the way. If the correct antigen is present, it binds to the antibody and continues flowing. The next molecule in the paper is in a stripe pattern. Should the third molecule bind the complex, a color change is seen by the naked eye. Antibody titers are done to determine the concentration of antibodies. It's done by diluting the patient's serum with equal amounts of the antigen. The highest dilution that produces agglutination or clumping is the titer. It's used for diagnostic purposes as well as to determine if a patient has been exposed to a particular disease. Studying antigen-antibody reactions can also be used to identify bacteria and place them into serotypes. It enables detection of certain virulence factors like a capsule in particular species and strains. The Western blot is used to determine the presence of certain proteins via an electrical charge. The gel is then transferred to a filter, which is incubated with antibodies. The antigen-antibody reactions can be visualized as bands, which help to determine the presence of specific antigens or antibodies in the sample. Here we see the basis of the reaction, with the target protein attached to the membrane blocks. The primary antibody binds to the antigen, along with the patient's antibody, which is a colorimetric label attached. These complexes can then be visualized as bands. Immunofluorescence testing employs the use of antibodies labeled with fluorescent dyes. In direct testing, the unknown sample is fixed to a slide with a known antibody. If the antibody is specific for the antigen in the sample, it can be visualized by fluorescence microscopy. It's useful for identifying microbes on cell surfaces and tissues, and can be used to identify Treponema pallidum, which is the causative agent of syphilis. This image shows a direct fluorescence test for the syphilis spirochete, which can be visualized by the fluorescence. In indirect testing, the fluorescently tagged antibodies recognize the patient's antibodies. This time, the known antigen is added to the sample. Binding can be observed through fluorescence microscopy, and this test can diagnose syphilis or other viral infections. In the ELISA test, which is an enzyme-linked immunosorbent assay, antibody-antigen interactions can be visualized. It relies on a microtiter plate that adsorbs or binds the reactant. An indirect ELISA is used to determine if particular antibodies are present in a sample. The known antigen is coated on the wells and mixed with an unknown antibodies from the sample. If the antigens and antibodies bind, a color change is seen. It's a useful test to screen for HIV antibodies, along with other viruses and bacteria. Sometimes false positives may occur, so a Western blot may also be necessary. In a direct ELISA, an antibody is coated on the well and incubated with the antigen in question. A color change develops if there is a reaction between the antigen and antibody. It can be used to detect antibodies to hantavirus as well as rubella virus. In the indirect ELISA on your left, the known antigen adsorbs to the well. The patient sample with unknown antibodies are added. The well is rinsed to remove unbound antibodies. An indicator antibody is added and binds to the patient's antibodies. The well is rinsed again and a colorless substrate is added. If it binds to the complex, a color change appears, but if no antibodies are present, no reaction takes place. In the direct sandwich ELISA on your right, a known antibody is adsorbed to the well. An unknown antigen is added, and if the antibody is specific to it, binding occurs. An enzyme-linked antibody is added, and it is specific for the test antigen. The substrate is then added, and if positive, a visible color change appears. Serological tests have high specificity and sensitivity. Specificity means that a test is specific for an antigen or antibody and does not react with any others. If a test has high sensitivity, it means that even in the smallest amount of antigen or antibody can be detected from a sample. Genotypic methods include PCR, hybridization, whole genome sequencing, along with PFGE. PCR, as you may remember, is like a Xerox machine that can make copies of DNA or RNA in a short period of time. Real-time PCR uses fluorescent tags that reveal amplification as it's occurring. It does not require analysis following the reaction. Not only can it identify a microbe, but it can also determine antimicrobial susceptibilities. Hybridization analyzes certain portions of a genome and assists in the identification of a microbe. Probes, which are small segments of nucleic acids, bind to the specific sequences that were isolated from a microbe. The base pairing can help in the identification process, along with color change. Fluorescent in situ hybridization, or FISH, uses probes attached to cells in a sample. Glowing cells can be used to determine the identity of a microbe and is useful in identifying a component in a biofilm. It's also used in the diagnosis of cancer and can be used to personalize treatment. This testing can be used to identify blood-borne pathogens quickly. A sample from blood is obtained, the probe binds to the sample, and the cells then fluoresce, which in this case indicates an infection with Staph aureus. Microarrays are also known as chips that can be used for infectious disease diagnosis. They contain gene sequences from a multitude of microbes. The patient samples are incubated with these gene sequences. The matching sequences bind to the chip, and a computer program determines the identity of the microbe. Everywhere that you see fluorescence indicates hybridization. Whole genome sequencing can be used in outbreaks as well as drug-resistant microbes. A single genome can be scanned multiple times, which helps to minimize errors. Eventually, this method will be so cheap and accessible that soon samples from patients can be immediately sequenced to determine the causative agent of the infection. Pulsed field gel electrophoresis separates DNA that your normal gel electrophoresis cannot. Voltage is applied in 3 directions, and restriction enzymes are used so that changes in the DNA sequence give rise to different sized fragments. This method can be used in outbreaks. There are genetic tests that have been placed on chips that don't require a significant amount of supplies or technical training. They can be used for DNA and RNA sequencing along with PCR. These tests are important in countries that have difficulty diagnosing infections due to lack of supplies, refrigeration, or medical personnel. Imaging can also be used for diagnostic purposes as well. There's a new strategy that checks a patient's blood for 7 genes that the host expresses in response to a bacterial infection. This will help in the treatment methods, as well as to decrease antibiotic resistance. There's also research being done to scan blood for antibodies to over 1,000 viruses.